Thanks everyone for being here today. I'm really excited to see the amount of people we have on and really excited to hear the work that's ongoing at the consortium today. As Michelle mentioned, Helen, our steering team member, was going to give this talk today, but she had a family emergency. So I'm going to step in. Some of you have heard this talk before, or at least a similar one. So I just want to give this today to get everyone on the same page and then also share a few high-level updates, but we will hear more updates today throughout the meeting. So I'm Connie Mitchell. I'm a scientific program manager at Hesse, or the Health and Environmental Sciences Institute. We are the convener of the consortium and really want to just set the stage today on why are we interested in botanical safety. So botanicals are complex. These plants are chemical factories. So there's around 30,000 that are reported to being used for medicinal use, but they're not really well studied. There's few cited in regulatory publications. And as many of you know, some of the secondary metabolites or phytochemicals can exhibit a broad range of bioactivities. And these constituents have really been utilized by humans for different reasons. So such as pesticides, pharmaceuticals, poisons, and other products. And there's a widespread use of botanical use through both the US and throughout the world. So this is a figure from ABC, but it just showed the amount of herbal supplement sales going up over time from the early 2000s to 2019. And then as many of you can imagine, supplement sales sharply rose during the first 6 months of the pandemic. And I believe that trend is ongoing. And there's this herbal use in supplements and products in the US, but botanicals are used all over the world. So it's used to treat different diseases in Africa and in Germany and Europe, and even in South America and the Americas. So it's not just in America usage of botanicals, it's really all over the world that people are turning it to either as medicines or as supplements. And there's inherent variation with testing plants or testing botanicals. So there's different steps in which the composition and the quantity of different constituents can change. So how is the plant grown? So the season, the soil conditions can influence that. How is it extracted? So what solvents used? Is there contamination? The finished product can be changed. So are there excipients, or is there a combination with other botanicals? And then how someone reacts to it can vary based on their own conditions, similar for single chemicals. But what's the exposure duration to the botanical? What's their genetic background? Are there pre-existing conditions, etc.? So all this makes it sort of difficult to test botanicals in a similar way as single chemicals, such as for pharmaceuticals. And that really leads into this slide. So testing schemes, especially in the US, but really around the world, are built around ensuring access to botanicals, whereas for drugs, what happens is the regulatory structures are typically aimed at ensuring safety and efficacy. A lot of times, pharmaceuticals are assumed harmful until they're proven safe. Usually the structures is simple and consistent, and we often know what the biological activity is associated with, which constituent, and what everything that is in the pill or however it's being taken. For botanicals, there's a lot of differences compared to pharmaceuticals. A lot of times the regulatory structures are aimed at ensuring access. A lot of times they're assumed safe until proven harmful. As I discussed, they're complex and variable. And sometimes we don't know what's driving the activity. And sometimes it's associated with the whole mixture, not just one constituent. So this makes it a little more difficult for toxicity testing and safety evaluation. So with these problems in mind, thinking that there's really limited tools to assess botanicals, a lot of times there's a reliance on history of use, and this really depends on regulatory body, but sometimes it's just built off of existing data that's already there or a history of people taking it. In addition, sometimes to fill gaps, often folks turn to in vivo testing to fill the gaps in knowledge on if something's safe or not. But this is costly, it's time consuming, there's ethical concerns. And what I just mentioned, that the variability, sometimes this isn't as useful. There's lack of global consensus regarding what compromises safety for a botanical. And sometimes safety requirements are diverse and varied depending what jurisdiction you're in or what context. And there's also little toxicity data on formulated products on the market. So some of the issues we talked about, but sometimes there's only toxicity data on a single constituent in the botanical, not the whole mixture. Sometimes botanical extracts and multi-ingredient products have no tox data. So maybe there's existing data on one type of extract of that botanical, but maybe not on an end-use product. And a lot of times approaches for single chemical entities don't fit for extracts. There's differences. They're not the same as single chemicals. So with these issues in mind, the Botanical Safety Consortium was formed a couple years ago. You might recognize some of the names on this paper, but basically groups came together at Eurotox in the ICSB meeting to really say, we need to do better. We need to have a cross-sector consortium to address this. And Scott Gottlieb, who was the former commissioner of FDA, actually announced that the agency was strengthening the regulation of dietary supplements by modernizing and reforming the oversight. And he announced the consortium as a public-private partnership that gathers leading scientific minds across the different sectors to promote scientific advances in evaluating safety of botanical ingredients in mixtures and dietary supplements. So that was in late 2018. Later that year, too, the FDA and NIH's National Institute of Environmental Health Sciences announced the official launch of the consortium with Hesse, the Health and Environmental Sciences Institute, as the convener. And really since early 2020, we've been meeting regularly and trying to progress the work. And that's really what you'll hear about today in some updates. And just 2 brief slides on Hesse. We're a science nonprofit based in Washington, DC. Our mission is to engage with scientists from academia, government, industry, and other scientific organizations to identify and resolve global health issues and environmental issues. And really, a lot of what I'll say about Hesse applies to the Botanical Safety Consortium, you'll see. And there's different ways we work on projects. So a few that relate to the Botanical Safety Consortium, it could be coming up with frameworks for how to test a certain substance. It could be working on data sharing and collective analyses, either with new experimental studies or with existing data. In all our projects, we publish everything and make it available, typically in the form of manuscripts. In some cases, we create tools or do assay development. So in this case for botanicals, do the assays that already exist work for botanicals as complex mixtures. And similar to the meeting we're having today and what you heard Michelle say about the different places we'll be to present our work, we attend scientific meetings, we have trainings, and we host meetings. And so our different focus areas are on there. Again, a lot of these apply to botanicals. So accurate and efficient chemical risk assessment, safe and effective medicines, environmental quality and sustainability, and food safety. And some of our other committees, all of our other committees are listed there, but I bolded the ones that most relate to the Botanical Safety Consortium, especially looking at toxicity endpoints. So for example, our developmental and reproductive tox group, our PBPK modeling group, and some of our other ones like our new tox group for different biomarkers there. And really, with this expertise in mind, we are able to manage the consortium, bring together our stakeholders, and bring in new folks that weren't initially a part of Hesse. So I'm going to talk about our current charge in the consortium. So with the problems I teed up and everyone coming together, really what we want to do is to evaluate the suitability of assays for botanicals as complex mixtures. So we want to be able to screen for toxicities to see what's going on in the botanical. Is there a flag? And here's our problem statement. So it's kind of just saying what I said in another way. So where we are right now is we don't know which new approach methodologies can be used to evaluate botanical safety. We have some ideas. There's been some great foundational work from different labs, from the National Toxicology Program, from academic labs, from companies, but still in a systematic way, we haven't evaluated which work for botanicals as complex mixtures. Where we want to be in a couple steps, and you see there's multiple arrows, is having a strategy and framework to evaluate botanical safety. So we want to know what tests work and be able to put them in a framework that others could use. And we have a couple objectives to do this. So first, similar to the big group you see online here, we want to connect with a broad group of global stakeholders to leverage the best scientific approaches. A big part in what you'll hear about today is the chemical analysis piece. So we want to be able to establish appropriate levels of chemical characterization for botanicals. And the other piece you'll hear a lot about today is the development of tools. So we want to come up with those pragmatic, fit for purpose, I'm going to say new approach methods or methodologies. So when I say that, I really mean non-rodent testing. So this includes in silico analyses, in vitro tests, and some non-protected animals like scaligons that we'll hear about later today. So really these quick tools that can be used to screen for toxicity that already exist for single chemical toxicity testing. We want to look at the assays and see how are they working? How do they compare to existing safety information? Do the tools pick up on the botanicals that we think to be positive? And really see, are these assays working? The ones we believe are working and maybe with some finagling integrate into that framework we talked about. That way others, the end goal being that others could be able to use that framework to try and screen botanicals for different types of toxicity or to see if they're safe. So with all these pieces, we want to be able to communicate our ideas. We want to have these presentations. We want to publish our results and then also present at conferences. So that's what we've been working to do and you'll hear about today. These are our different toxicity endpoints, or excuse me, these are our different themes throughout the consortium, including our toxicity endpoints that are seen on the left. So you can see the ones listed there: hepatotoxicity, cardio, neurotox, gene tox, developmental and reproductive. And then one group to kind of tie them all together, our systemic toxicity group. These were identified as our initial endpoints. It doesn't rule out looking at other ones, such as maybe renal or skin sensitization in the future, but this is what we're getting started with. And really, since we're looking at some of these new approach methods, it's really important to know the differences between exposure from a in vitro system versus, a human or even an animal. So absorption, distribution, metabolism, and excretion are really important. So that's a big part of what we're looking at and how to interpret the results of an in vitro test or an in silico assay. Another big thing we're interested in is botanical drug interactions. So how can if you're taking a pharmaceutical, how could a botanical potentially affect that? And what tools are there out there to look for those interactions? We have a chemical analysis group that's really instrumental, along with a data analysis group to help us interpret results. Another theme that's really important is that in vitro to in vivo extrapolation. So how do the concentrations we're testing in vitro compare to what's being reported? What's going on there with some of the ADME characteristics and how that fits in. And one group that spans all these different topics is our pharmacognosy group. So really, how are these plants or botanicals taken either traditionally or currently? How do we source them? What are the known constituents that are either used for, for the pharmacoactive piece or for the toxicity piece? So really bringing all these groups together, and some of which that haven't been brought together before to look at botanical safety has been really great. And just having different minds and different backgrounds evaluate these different topics. We also have literature reviews for our botanical case studies. So gathering that existing information to try and compare and tie our in vitro and new approach methodology results to. And here's our strategy of how we're trying to get to that framework. So if you see on the far right, so as I said, we're trying to develop the framework that has a toolkit of different assays in it. And really how we've started is we had our different toxicity endpoints. And the work we've been doing the past 2 years is sort of twofold. So looking at this figure in the middle at the top part, the different groups identified botanical candidates. And these are botanicals with either known safety or toxicity concerns. And these aren't necessarily botanical supplements. Some of these are plants that have been off the market or not allowed to be taken as supplements. But really, what are the positives for the different endpoints that are known? So the groups looked through the existing literature and selected botanicals that were known to be, like I said, either assumed positives or negatives for those groups. We obtained large amounts of all the different botanicals that we could. And what we've been working on and what you'll hear about some updates today is making sure we're testing what we think we're testing. So we've done a few different methods to make sure what we're testing has the right constituents. If there's any toxicity, it's not being driven by something that's not supposed to be there or some kind of misidentification. So we did a lot of work on the chemical analysis side to compile our libraries. At the same time, if we're now looking at this figure at the bottom part, the different toxicity groups identified assays and approaches that already work for single chemicals. We worked with both the chemical analysis team and data analysis team to design the studies. And what we're starting to do, and you'll hear about today, is run the botanicals we compiled through those assays. What we're going to be working on in the next couple months to years is trying to see, okay, we ran the assays, how does it compare within the assay itself? And then how does it compare to the existing data? Is that assay, understanding it has limitations and context of use, but can it be used as part of a toolkit to screen for toxicity? So we're working on all these steps to then get to that framework. Throughout the process, we have ongoing stakeholder communications and engagement. So in meetings like this, in sessions like the Society of Toxicology or the ICSB meeting, or just having people be able to write in and tell us and give us feedback. I'll go through this briefly. This is how we came up with selecting our botanical candidates and all the work we needed to do before we could actually start testing them in the assays. But really, like I mentioned, in those toxicity groups, we were looking to see what botanicals are already known to induce those types of toxicity. So we gathered information on the different types. So it could have been in vivo evidence, like a study in rodents. It could have been human evidence, so adverse event reports or clinical trials. Some of it might have been mechanistic or in vitro. There were some factors that weren't the deciding factors, but helped us in narrowing down. So are they used in susceptible populations? So example for development and reproductive tox, are there certain populations that are more vulnerable? And also, were there known toxic constituents? So we're really looking at well-studied botanicals here, not unknowns. And the different groups selected really a long list of positives. What we'll see here today is we wanted to just get started. This has been a long process already. So we selected 13 that I'll show on another slide that cover a lot of these different topics here. And what we had to do that I sort of mentioned for the 3 different steps in the middle, really we wanted to make sure we're testing what we think we're testing. So we wanted to do both authentication and look at what's in there. So the constituent identification and quantification. We also wanted to gather the existing information. So the groups gathered that information shown on the left, so the different evidence types, but also information on how the botanical is used. What are the known constituents? What other information would be relevant in assessing an assay? So we've gathered information on our case studies, and we've also had a lot of help from the pharmacognosy team. And so they helped us identify sources and suppliers. So everything we're looking at right now is not off the shelf. It's from manufacturers or growers. So they helped us get in touch with those different manufacturers or growers to get large amounts of the botanicals so we could test the same lots in all the assays and in the chemical analyses we did. They also helped us gather some information that fed into the lit reviews. So adverse events, indications of use, what are some of the doses? So that'll help us tie what we're seeing in the in vitro studies to what's actually going on when someone takes these, some of the known contaminants or adulterations, and the nomenclature. So really, we've been working on this the last 1.5 to 2 years to get all this information together. And so once that was all together, we've now been able to begin our assays. So you'll hear about today some of the assays we're really just now getting started on. And in the next year, we'll really present those results of which assays are potential being able to be used in a toolkit. So I like this slide because I just talked for quite a while about what we are doing, but I think this slide is nice because it says what we're not doing. So we're not making safety determinations on the botanicals. We're really focused on the assays. We want to say what assays can be used to screen for the different toxicities we're interested in, but we're not trying to say botanical X is safe or this one's toxic. We're not making regulatory decisions. We're not endorsing any particular botanical product. Right now, we're not testing anything off the shelf. These are really botanicals that we got large amounts of that we can try and evaluate the assays with. We're not looking at just the individual constituents. So we're looking at botanicals as the whole mixture. In some cases, the assay itself may be conducive to have maybe one constituent compared to the complex mixture, but we're really focused on the complex mixture. Currently, we're only looking at the oral route of exposure. That's something that once we have an existing toolkit could be expanded, like I mentioned, maybe to skin, maybe to inhalation, but really we want to start with these foundational work. And a lot of what we've done here would apply to the other routes of exposure, especially considering different ADME tools and ideas there. We are not performing mammalian in vivo studies, so we're not doing rat or rodent studies. In some cases, what we might find is there's not currently an in vitro or new approach method to cover that endpoint. So we might find that and say that as a gap analysis, but we ourselves are not performing those mammalian studies. And similarly with the safety determination, we're not doing risk assessment. So we're not, again, we're really not focused so much on the botanical, we're focused on the tools you could use to evaluate it. So I hope that helps with some questions. There's just a slide on assay guidelines. So how did we select our different assays? We wanted ones that are established and reproducible. We wanted them to be for key mechanisms of botanical induced toxicity. We wanted them to be accessible by many groups, so not something proprietary. We need them to be suitable for botanicals complex mixtures. So there's differences between testing just a single chemical in the mixtures, and cost was also something we're considering. And we're going to hear more about these assays today. Cynthia is going to go through a few of these points of why we're doing this again later on. And I just want to show this briefly. You'll hear about some of this today, but these are the ones we're initially trying. So not all of these might not be suitable. There might be limits on when they could be used, but these are the ones we're getting started with of the different groups selected. And I'll go through just a few highlights. So some have already been tried for botanicals quite a bit through different programs. So like the Ames test and the in vitro micronucleus. There's others that haven't been worked out as much or maybe haven't been tried for botanicals. And there's some that also cover multiple endpoints. So for example, we have transcriptomics, both in the DART group and the hepatotox group, but that will provide information on other endpoints and could help narrow in on potential mechanisms. There's also some of the non-rodent, non-protected animals, so such as zebrafish embryos, which can give us both information on development and reprotox, in addition to neuro and maybe even some info on cardio or other endpoints. And there's some, if you see the hepatotox as a long list, but a lot of these can be complex together. So that's also a consideration for both the time, cost, and resources. So you're going to hear quite a few different assay talks later today. So I won't go into too much more detail, but like I said, these are the ones we're getting started with and trying, and we should have more results in the next couple of months. Here are our different botanical case studies. This is another one. I'll share a few examples. We can talk in detail on why these were chosen, but these were the ones chosen by the different working groups for either their known toxicity or a certain factor or safety. So for example, comfrey is one that contains PAs, so it's known to be genotoxic, and it's not taken as a supplement, especially not in the US, it's actually banned. There's a few others on here, so say green tea. What we're testing is the green tea concentrated extract that's decaffeinated. And this matches a lot of existing data that's already been done at the National Toxicology Program. And there's a lot of information that that specific one, not all green tea, but that specific extract can lead to hepatotoxicity and botanical drug interactions. So on here too, there's nuance in which one we selected. So it's not all green tea, it's just the one specific. Some of our pan negatives, that have a lot of history of use and well-established safety in clinical trials and different data are Asian ginseng and milk thistle. And then other ones were chosen for different groups or might go across different groups. So for example, ephedra is one we're looking at both in our neuro and cardio groups based on existing information. It's another one that has been banned in the US, so it's not something currently on the market. So just for sake of time, we have information on why we selected each of these. We won't go into too much more detail today, but if there's questions, Michelle and I can take that later. I just want to walk through how we foresee the example assays going. It's something we'll hear about more with the assays today. But basically, we'll have different treatment groups. So it really depends on the assay, but there might be chemical positive controls, chemical negative controls that are specific to that assay. And then our botanical proposed positives, our botanical proposed negatives. And as the assay allows maybe other botanicals in there as well that don't have reported data for that endpoint. Let's say it's a 96 wells in vitro assay. We would expose the cells to the botanicals for the appropriate exposure amounts and duration. We would measure the endpoints. We would analyze the data in the assay itself. So how did it compare to the controls? How did it compare with everything? But then we're also going to compare to our existing toxicity data. Does it line up? Did we get what we expected? Why not? Were there certain considerations that we didn't think of, like solubility or some kind of fluorescence issue? We'll refine and retest as needed. And then the ones that are working, we will integrate into our framework. And some anticipated applications of the framework. So what could others use once that framework's available? So you could screen new or data poor botanicals for safety. You could screen and compare products on the market. So maybe lot to lot comparisons. You could prioritize botanicals for animal testing, or conversely deprioritize the need for some animal testing. So really thinking about the 3 R's here. And also to fill knowledge gaps. So some of these tools will give you information on mechanisms. So if it is causing toxicity, how? So those are some anticipated applications of the framework we see people could be able to use. I want to go through just a timeline of what we've done since we've really gotten started in early 2020. As you know, it's been difficult. Everything's been slower since the pandemic, but we've really made a lot of progress over the last 2.5 years. So in early 2020, we formed our working groups and started meeting regularly. Throughout 2020 and 2021, we selected our botanical case studies and assays. And in 2021, we started obtaining materials and beginning some of our chemical analyses work and the in silico work. So early this year, we finalized our assays and we got going on the rest of our chemical analyses. We're finishing up for the botanical case studies. This summer, we started some of our assays to see if they're suitable for botanicals. And this fall, what we're doing is we're starting to look at the results of those assays. We're looking at the in silico analyses, we're looking at and continuing the testing. Next year and later this year, we foresee maybe some publication of initial results, or at least presentations of how things are going. Next year, we'll continue testing as needed and also maybe do some retesting if there's issues that come up. There's potential to do other work like test more botanicals. So right now we have just those 13, but we might expand and see the different context of use of the assays and really work on refinement of that toolkit and framework. Future phases, I think there's a lot of work to do on every call. I feel like we come up with work we're going to do in phase 3, 4, 50, but there's some just ideas listed on the screen. So there might be other endpoints, like I mentioned, like skin sensitization, renal, maybe comparing sufficient similarity. This is work that groups are doing, especially the National Toxicology Program. So how do the chemical differences in botanicals translate to the biological differences you see in the assays? And there's also potential follow-up. So a lot of what we're looking right now is screening tools, but there's also mechanistic assays that could really identify how the toxicity is occurring. So that might be of interest if you're really doing a deep dive on a botanical, or if you think an assay could capture why this is happening or what's the actual mechanism it's working through in a person. So there's a lot of different work we can do in the future. Just briefly, before I end today, talk about some of the outreach we've done. So this is just from maybe the last 1.5 years, but we had a session virtually at the SOT 2021 meeting. They also had a keynote at the Congress of Toxicology and Development, in developing countries. We had this year a platform at the German Farm and Tox Summit. This year at SOT, when it was in person, we had 4 posters from our different working groups and a session at ICSB that a lot of our steering team attended and spoke at. We've had one of our steering members, Olaf, has been really great about promoting the Botanical Safety Consortium at meetings around Europe. So we had a poster at the Phyto Congress meeting this year. Our Gene Tox group and other working groups have been really great at presenting the work they're planning or have already piloted. So we had at the environmental mutagenesis meeting this past August, had a platform given by Stefan Fuller. We had a workshop at the GA meeting. So that's the meeting that just happened in Greece. So that's a more phytochemical meeting, but we had a workshop on safety preceding the meeting that was really well taken up. And that's probably a meeting we'll target in the future. So myself and Michelle were there, along with quite a few of our working group leads or steering team members. We just had a poster at the Eurotox meeting. And as Michelle mentioned, some of the upcoming events, our Gene Tox group again is going to meet at a German Gene Tox meeting next month. And we also have a session for SOT 2023. So like I said, we're trying to get the word out there, and we'll be at some other meetings in person. So if you're going to those, just let us know and we'll try and meet up. Briefly want to talk about some of the global outreach we've been doing. So throughout the work we're doing within the consortium and coming up with the toolkit, we've been able to meet a lot of really great people and come up with some other ideas of smaller projects that we've been working on. So 2 are going at the Kalamazoo University of Health Sciences in Malawi. One was a survey of herbal drug interactions. So we really looked to see what people at a clinic are taking in terms of botanicals or herbal medicines, and then what other conventional medicines are they taking. So this was performed with researchers at that university. We collected the data and we're doing an analysis now with those collaborators. And then we also, from that work, a smaller project came out to look at different Aristolochia species that are traded and taken in Malawi. So really to quantify to see if there's the different aristolochic acids in different Aristolochia species that people are taking for medicine in Malawi. So just wanted to mention these 2 projects. I think they're really exciting, even if they're not our main focus. And we have a lot of work that's coming out of it that we'll hopefully be able to present in the next couple months. So just acknowledgement to the team there, especially the folks at the Kalamazoo University and our pharmacognosy team that's really been helping with this. Just a couple publication updates. So early this year, we had a paper that really just went through the things I just said. So what's the consortium doing? What's our strategy? And some of the initial case studies we selected and why. So that's out in Rake Toxin Farm, available open access. We also were invited to write a paper on nephrotoxicity of botanicals. So if you notice, that wasn't one of our groups, but really just with the expertise we had in the consortium, and we found a really motivated postdoc at the National Institute of Environmental Health Sciences to help us write that. So similar to what we're looking at the consortium, we did a small review of what has been reported on botanicals that lead to nephrotoxicity. We found there was really big mix in the data that's out there. So what we did is we recommended tools that you could use to screen for nephrotoxicity. We have quite a few different manuscripts in progress. So for ashwagandha, one of our botanical case studies, we were able to do a deep dive into that with our colleagues at Procter & Gamble and other people at the in our chemical analysis group. So we're writing that up, and that should be available maybe next year. We're also informing our literature reviews. We came up with a list of recommended information and sources of botanical toxicity studies. So really information and authoritative sources you could use if you're a toxicologist running a botanical study. So what's the work you need to do first to gather the relevant information? What we found is in some papers, there's really not a lot of information on what people are testing. So really, what information do you need to include to make it a more robust study? So that's going on within our systemic toxicity group. Develop Patel is helping to lead that, along with Stephen Nantali and others in that group. So we're hoping to get that out in the next couple months, maybe early next year. And then some of the work from the different Malawi projects, we're working to get that out as well. And there might be some other papers coming in the next year to couple months. So some of the work in the chemical analysis group that needed method development. So for example, for kratom and for blue cohosh. And in our ADME team, we've done a lot of PBPK modeling. So that's something else we're trying to package and put out there. So be on the lookout for more papers in the next year. And I think that's all I have today. Checking on time, maybe 1 or 2 clarifying questions we have time for. Michelle, do I need to unmute you before we turn over to Victor? Let me just see if there's,
I don't see any questions in the chat. I did want to bring up, Joe Betts brought up a very, very good point related to existing information that might be available from veterinary studies for animal poisonings. And he sent a link, and I think that's a really good addition to some of the places that we might look for existing toxicity information for plants.
Stefan asked if the posters are available on our website. That's something that we definitely can do, especially for, we have a general one on the BSc that's a nice snapshot, and some of the ones we had from SOT. So if they're not, I can make them available. That's a great point, Stefan. Thank you.
I think they might be on our like our internal site, but not on the public site, but we can put them on. That's great.
There's a question on curcumin. So again, we're really focused on what tools we can use to evaluate it. So it's not something we've done a deep dive into, but thank you for that question. That might be something good to keep on our radar. Once we have a functioning toolkit, that might be something to look into, or someone could take up to look into that with the tools we have.
Connie, I was also just going to say, I think, you know, largely because this is a community of practice, even though we aren't addressing particular questions about the question on curcumin, if there's others on this consortium who might reach out to the person who asked that question and could sort of take that offline and address that outside of the BSc, feel free to respond directly to Mithili in the chat, because we do have a large group of people here who might be able to address that.